Hi there, my name is Vinay Pathak, and this is my first Advanced Concepts presentation for Biochemistry 1. Today we're looking at the leucine zipper. For prompt 1 it says, explain what principles are involved in holding the two alpha helices together. Suggest a possible residue that could replace leucine and maintain the hydrophobic core shown below, and suggest another possible residue that would destabilize the core. So obviously protein structures can be held together by a number of different forces, but looking at the diagram here, the two forces holding the alpha helices together are potential electrostatic attraction and hydrophobic core interactions. For the potential electrostatic attraction, that is lysine and glutamate that are attracted to each other. And in this case, at the physiological pH, lysine is an ammonium ion. So the nitro group on the end of it, on the side chain, is protonated, which makes the whole entire molecule positive. And for glutamate, at the physiological pH, the carboxylic acid side chain on the end is obviously deprotonated, and so it's negative. And so the ammonium ion, which is lysine, and the negative glutamate are attracted to each other, which helps hold together the two alpha helices. For the hydrophobic core interactions, obviously all of the amino acids that are not participating in the potential electrostatic interaction, they have side chains that are nonpolar, and so those will make up the inner part of the leucine zipper and interact with each other, creating a hydrophobic core. And that is important because when nonpolar side chains interact with each other, van der Waals or dispersion forces occur, which are weak forces of attraction, but they do play a role in holding the two alpha helices together. For the residue that would maintain the hydrophobic core, I chose isoleucine, and that's an obvious one because it's a constitutional isomer of leucine. So because it's a constitutional isomer, it has the same atoms in the side chain, just arranged differently. And so it's going to exhibit the same level of hydrophobicity. And so if leucine was replaced with isoleucine, it would maintain that hydrophobic core. For residue that would destabilize the core, obviously the hydrophobic core, that would be thrown off by threonine, and that's simply because it has a hydroxyl group. And so due to this hydroxyl group, it makes the side chain hydrophilic. And obviously a hydrophilic side chain doesn't cooperate well with hydrophobic side chains, and so that inner part, the hydrophobic core, would be disrupted because the hydroxyl group would naturally gravitate and be attracted towards the outside because it wants to interact with water. So to sum up prompt 1, the principles involved in holding the two alpha helices together are potential electrostatic attraction and hydrophobic core interactions. The residue that would maintain the hydrophobic core is isoleucine, and the residue that would destabilize the core is threonine. So for prompt 2, it says to propose a different amino acid pair whose side chains could interact in a similar manner to lysine and glutamate. So just to recall, lysine obviously was the ammonium ion here, and so it was positive, and glutamate had the negative carboxylic acid side chain, which was negative, and so obviously the positive and negative played a role in holding the two alpha helices together through that potential electrostatic interaction. So in this case, I'm looking for an amino acid that would be negative at the physiological pH of around 7. And so I would look for an amino acid that is more acidic. And one of the ones that I thought would work would be aspartate or aspartic acid, and that has a 3.9 pKa, and at the physiological pH, it would also have that carboxylic acid group being negative. Obviously, the aspartate was to substitute in for the glutamate, but in terms of what substitutes in properly for the lysine, it would be something that is positive at a physiological pH of around 7. And so I arbitrarily selected arginine. And so arginine is obviously basic. It's basic relative to physiological pH. It has a 12.48 pKa. And so because of that, it's positive at physiological pH. The guanidinium group would obviously be protonated, making it positive. And because it's positive, it could have that same potential electrostatic interaction with aspartate. And so that would help hold the two alpha helices together. So just to sum up prompt 2, propose a different amino acid pair whose side chains could interact in a similar manner to lysine and glutamate. For lysine, I selected arginine to be positive at the physiological pH, and for glutamate, I selected to substitute in aspartate, which is negative at the physiological pH.